AI compared the DNA of every human race on Earth simultaneously, something no human researcher has ever been able to do. And buried inside that data is a pattern that keeps appearing across populations that should have never met. Same marker, different continents, different eras. No explanation. Today we walk you through what the AI found and why 3 research teams are currently scrambling to explain it. Every human population on Earth descends from African ancestors. That is not a theory. It is the most replicated finding in the history of genetics, confirmed by mitochondrial DNA analysis, Y chromosome haplogroup mapping, and full genome sequencing across hundreds of studies. But knowing that all humans came from Africa is different from understanding what Africa's genetic landscape actually looks like and what it reveals about human origin. The AI's first task was establishing the baseline. Before it could compare populations to each other, it needed to understand the full range of human genetic diversity. And that range, it turns out, exists almost entirely within Africa. Here's the finding that most people do not know. The genetic distance between 2 randomly selected individuals from sub-Saharan African populations is greater than the genetic distance between a West African and a Northern European. Read that again. 2 people from different African ethnic groups, say a Yoruba from Nigeria and a San person from Botswana, are more genetically different from each other than a Yoruba person is from a Swedish person. The Khoisan are not more primitive, they are more ancient. Their lineage is a direct window into what human genetics looked like before our species began the migrations that eventually covered the entire planet. The AI mapped 5 distinct genetic clusters within Africa alone, each representing populations that had been genetically separate for tens of thousands of years before any human left the continent. West African populations, the Yoruba, Igbo, Mandinka, formed one cluster, characterized by Y chromosome haplogroups, E1b1a, and mitochondrial haplogroup, L2 and L3. East African populations, Amhara, Somali, Maasai, formed a second cluster, with significant haplogroup E1, B1b representation, and mitochondrial L, 3 variants that sit closest to the out-of-Africa founding lineages. Central African forest populations, the Aka and Baka pygmy groups, formed a third cluster, with genetic divergence times from other African populations exceeding 60,000 years. Southern African Khoisan populations formed the 4th cluster, the oldest, most ancestrally distinct of all, and a 5th cluster emerged that surprised the research team, a genetically intermediate group spanning the Sahel and Horn of Africa, showing significant admixture between sub-Saharan African populations and ancient North African and Middle Eastern populations, a signal of back migration into Africa that occurred approximately 3,000 years ago and left detectable traces across a wide swath of East Africa. So Africa is not a single genetic story. It is 5 overlapping stories, each older and more complex than anything that happened after humans left the continent. Between 50,000 and 70,000 years ago, a small group of humans left Africa. The AI's analysis placed the founding population at between 1,200 and 2,500 individuals, an extraordinarily small group to serve as the genetic ancestors of every non-African person alive today. This bottleneck is visible in the data. Non-African populations showed dramatically reduced genetic diversity compared to African populations, exactly what you would expect if they descended from a small founding group that carried only a fraction of Africa's genetic variation. The exit point was almost certainly somewhere along the Arabian coast, modern-day Yemen or Djibouti, where the distance between Africa and Arabia narrows to just 30 kilometers at the Bab el-Mandeb Strait. Those first migrants spread rapidly. The AI's divergence calculation showed that the founding population split into multiple branches within just a few thousand years of leaving Africa. One branch moved along the southern coast of Asia, island hopping through what is now India, Southeast Asia, and eventually reaching Australia and New Guinea, approximately 50,000 to 65,000 years ago. Another branch moved north and west, eventually producing the populations that settled the Middle East, Central Asia, and Europe. A third branch moved northeast through Central Asia and into Siberia, eventually crossing the Bering Land Bridge into the Americas. But the AI found something embedded in this dispersal that standard migration models had underrepresented. These branches were not clean separation. They overlapped, they merged, they split again. The populations that eventually settled Europe were not simply African migrants who walked north. They were the product of at least 3 distinct waves of migration into Europe, each genetically distinct, each contributing differently to the modern European genome. And the populations that eventually became East Asians were not a single coherent group either. And the AI detected at least 2 distinct founding lineages contributing to modern East Asian ancestry, a northern lineage associated with the Yellow River Basin and a southern lineage associated with coastal Southeast Asian population that had been partially separate for over 20,000 years before the agricultural expansions of the Neolithic Paleolithic merged them. The clean arrows on the migration maps and history textbooks are wrong. Human dispersal was messy, recursive, and far more complex than any simple diagram can convey. When the AI turned its full analytical power to European populations, it produced findings that fundamentally challenge the way Europeans think about their own ancestry. Modern Europeans are not the descendants of the people who first settled Europe. They are not the descendants of the ancient hunter-gatherers who painted the caves at Lascaux and Altamira. They are not primarily the descendants of the early farmers who built Stonehenge or the megalithic tombs of Brittany. Modern Europeans are, genetically speaking, a recent hybrid, a mixture of three distinct ancestral populations that only came together in their current proportions within the last 5,000 years. The AI identified these three components with high precision. The first component, Western hunter-gatherer ancestry, traces back to the Paleolithic populations that moved into Europe after the last glacial maximum approximately 14,000 years ago. These people carried Y chromosome haplogroup 1 and mitochondrial haplogroups U4 and U5. And their genetic contribution to modern Europeans ranges from 10% in populations near the Ukraine to roughly 25% in Scandinavian populations, where hunter-gatherer ancestry persisted longest. The second component, early European farmer ancestry, derives from Anatolian farmers who moved into Europe approximately 9,000 years ago, bringing wheat and barley agriculture with them. They carried haplogroup G2A on the Y chromosome and mitochondrial haplogroups H, J, and T. Their genetic contribution ranges from 30% in British and Scandinavian populations to over 60% in Sardinian populations, Sardinia being the region where Anatolian farmer ancestry was most preserved and least diluted by later migration. The third component, and the one that most dramatically reshaped European genetics, was the steppe ancestry. In pastoralists from the Pontic Caspian, steppe north of the Black Sea, ancestral to the people who spread Indo-European languages across Eurasia, expanded into Europe approximately 5,000 years ago, carrying Y chromosome haplogroup R1b in the west and R1a in the east. The AI calculated that this steppe expansion replaced over 75% of the male lineages in Britain within just 300 years. The Bell Beaker culture, which spread across Western Europe between 4,700 and 3,800 years ago, was genetically associated with this steppe ancestry, and its arrival in Britain corresponded with one of the most complete genetic replacements ever documented in a large population. The people who built Stonehenge were almost completely replaced by incoming steppe migrants within a few centuries of the monument's completion. Now, modern Europeans carry these three ancestries in varying proportion, depending on geography. The gradient is precise, and the AI mapped it at high resolution. Northwestern Europeans, Irish, British, Scandinavian, carry the highest steppe ancestry, typically 40 to 50%. Southern Europeans, Sardinians, some Greek populations, parts of the Balkans, carry the highest early farmer ancestry and the lowest steppe ancestry. Eastern Europeans show the highest proportions of both steppe ancestry and residual hunter-gatherer admixture. No modern European population is more than 50% genetically continuous with any population that lived in the same geographic area more than 5,000 years ago. When the first humans crossed from Siberia into the Americas, either across the Bering Land Bridge or by boat along the Pacific coast, they entered a world that no human had ever seen. They also entered genetic isolation. For approximately 15,000 years, the populations that filled North and South America evolved with almost no genetic input from the rest of humanity. The ocean barriers on both sides and the retreating ice sheets that closed the Bering Corridor after the initial crossing created the most complete population isolation in human prehistory. The AI's analysis of Native American populations reflected that isolation clearly. The founding population of the Americas traces to a single ancestral group that the AI estimated at between 250 and 1,000 individuals, an even more extreme bottleneck than the one that produced all non-African diversity. This founding group crossed into the Americas carrying Y chromosome haplogroup Q, specifically Q1A2A1, and mitochondrial haplogroups A, B, C, D, and X. Those 5 mitochondrial haplogroups account for over 97% of pre-Columbian Native American maternal lineages. The genetic diversity within the Americas is extraordinary in terms of language and culture, but genetically, all Native American populations share a common origin point that is far more recent and far more restricted than the origin points of populations in Africa, Asia, or Europe. The AI found that North and South American populations began diverging from each other approximately 13,000 years ago, consistent with a rapid dispersal southward through the Americas after the initial entry through the Bering Corridor. But it also found something more specific about the founding event. The ancestral Native American population was not genetically homogeneous, even at the moment of entry. The AI detected 2 distinct genetic signals within the founding group, a primary signal matching Siberian ancient North Eurasian populations, and a secondary signal matching a population more closely related to ancient East Asian population from coastal China and Japan. The Americas were not founded by a single coherent Siberian population. They were founded by a mixed group, part Siberian steppe heritage and a part ancient Pacific Rim coastal heritage that had merged somewhere in Beringia before pushing into the New World. That coastal Pacific signal explains something that has puzzled geneticists for decades. Certain South American populations, specifically groups in the Amazon basin and along the Pacific coast, show genetic signals faintly reminiscent of Australasian population. The AI's analysis supported the hypothesis that a second, earlier wave of migration, carrying Australasian-related ancestry, preceded the main founding population and left traces in the deepest layers of South American genetic history. Two waves, not one. And the second wave was almost completely absorbed by the first, leaving only faint traces in the most geographically remote South American populations. If the AI found one region that defied every attempt at simple categorization, it was South Asia. The Indian subcontinent is home to over 1.4 billion people, spread across thousands of distinct ethnic communities, caste groups, and linguistic population. And the AI's analysis revealed that this cultural complexity reflects genuine genetic complexity, layered migrations, ancient populations, and admixture events spanning the entire history of modern humans in Asia. The AI identified 4 distinct ancestral components contributing to modern South Asian population. The first, Ancient Ancestral South Indians, or AASI, represents the original hunter-gatherer population of the subcontinent. These were the descendants of the first out-of-Africa migrants who moved along the southern coastal route into South Asia over 50,000 years ago. They carried mitochondrial haplogroup M, a lineage that diverged from the African L3 founder approximately 65,000 years ago. Their Y chromosome haplogroups are difficult to resolve cleanly, but include deep branches of haplogroup H that show no close relatives outside South Asia. AASI ancestry survives at highest concentration in certain South Indian tribal population. The Jarawa of the Andaman Islands show the closest modern genetic parallel to the ancient AASI population, having been isolated on their islands for over 30,000 years. Their genetic profile is a living snapshot of what the first South Asians looked like before any subsequent migration. The second component, Indus Valley Civilization ancestry, derives from the urban farming populations of the northwestern subcontinent who built Harappa and Mohenjo-Daro between roughly 5,500 and 3,900 years ago. Genome sequencing of Indus Valley skeletal remains revealed a population that mixed AASI ancestry with early Iranian farmer ancestry, itself a mixture of ancient Iranian hunter-gatherers and early Anatolian-related farmers who moved east across the Iranian plateau. The Indus Valley population contributed significantly to the genetic profile of modern South Asians, particularly in Pakistan and northwestern India, where it accounts for 45 to 65% of ancestry in some population. The third component, steppe ancestry, arrived in South Asia approximately 4,000 to 3,500 years ago, i.e. carried by pastoralists from the Pontic-Caspian steppe, who also swept into Europe during the same period. These were the people who brought Indo-European languages to both Europe and South Asia. Their Y chromosome haplogroup R1A, specifically the branch R1A-Z93, shows up at high frequencies in upper caste South Asian populations and in populations across Central Asia and Iran. The AI found a clear correlation between steppe ancestry and social position in the ancient Indian caste hierarchy. Upper caste populations in northern India show significantly higher proportions of steppe-derived R1A than lower caste and tribal population. The genetic stratification of the caste system is visible directly in the haplogroup data, while the fourth component, East Asian ancestry, appears in northeastern Indian populations and increases in frequency as you move toward the borders of Myanmar, China, and Bhutan. These northeastern groups carry Yellow River-associated haplogroup O lineages at rates of 30 to 60%, reflecting the same agricultural expansion that filled East and Southeast Asia over the past 5,000 to 7,000 years. 4 distinct ancestral populations, contributing in different proportions to thousands of different communities across the subcontinent. South Asia is not one genetic story. It is 4 ancient stories that collided in one place and and never fully resolved. The Fertile Crescent, the arc of land running from the Nile Delta through the Levant, down through Mesopotamia to the Persian Gulf, is where agriculture began, where writing began, where the first cities appeared. It is also, genetically, one of the most churned and complex regions on Earth. The AI's analysis of Middle Eastern populations revealed a landscape that looks almost nothing like it did 10,000 years ago, even as the region sits in the same geographic location where civilization first emerged. The founding Neolithic farmer populations of the Fertile Crescent carried Y chromosome haplogroup G2A and mitochondrial haplogroups T, J, and H. These were the people who domesticated wheat and barley, who built Jericho and Çatalhöyük, who eventually sent agricultural migrants into Europe, South Asia, and North Africa. The AI calculated that direct descendants of these founding Neolithic farmers account for fewer than 25% of modern Middle Eastern ancestry. 3/4 of the modern Middle Eastern genome comes from populations who were not there when civilization began. The first replacement came from steppe-related pastoralists moving through the Caucasus and into the northern Fertile Crescent, approximately 4,500 years ago. They brought bronze metallurgy, horses, and ultimately contributed to the genetic profiles of populations across Iran, Turkey, and the northern Levant. The second came from Arabian Peninsula population, expanding northward during the Islamic period, post-7th century CE. This expansion brought haplogroup J1, the dominant Arabian Y chromosome, into populations across a vast geographic range, contributing significantly to Egyptian, Levantine, and Mesopotamian male lineages. The third was the continuous back migration from North Africa, populations moving north across the Sinai, that has occurred repeatedly throughout history and introduced sub-Saharan African admixture into North African and Levantine populations, at rates ranging from 4% in Lebanese populations to over 30% in some Egyptian communities. The people living in Iraq today are not genetically continuous with the Sumerians who built the first cities in the same location 6,000 years ago. The people living in Egypt today carry only partial genetic continuity with the ancient Egyptians whose mummies fill the world's museum. The birthplace of civilization has been genetically overwritten multiple times. When the AI reached the Oceanian populations, Aboriginal Australians and Papua New Guineans, it flagged a series of findings that it classified as requiring independent verification due to their statistical outlier status. These were not errors. They were the genuine genetic signature of the oldest populations outside Africa. Aboriginal Australians and Papua New Guineans descend from the first wave of out-of-Africa migration, the coastal route that moved east along the southern rim of Asia, approximately 50,000 to 65,000 years ago. They split from all other non-African populations before the ancestors of Europeans, East Asians, or Native Americans had differentiated into distinct groups. The genetic distance between Aboriginal Australians and any other non-African population is greater than the genetic distance between any two other non-African populations. Aboriginal Australians are genetically closer to the out-of-Africa founding event than any non-African population except possibly certain Andaman Islander groups. Their mitochondrial haplogroups, M and N lineages, in distinct ancient configurations represent branches of the founding out-of-Africa lineage that diverged before the lineages producing all East Asian, European, and Native American population. But the AI's most significant finding about Oceanian populations was not about their ancient ancestry. It was about Denisovan admixture. Denisovans, the archaic human species known primarily from a finger bone found in a Siberian cave, interbred with modern humans at multiple points. The Tibetan EPAS1 variant is one legacy of that interbreeding, but the primary Denisovan signal in the modern human genome exists in Oceanian population. Aboriginal Australians and Papua New Guineans carry approximately 4 to 6% Denisovan ancestry, significantly more than any other modern population. East Asian populations carry roughly 0.2% Denisovan ancestry. European populations carry essentially none. The AI mapped this distribution against the known geography of Denisovan population, which, based on ancient DNA and environmental DNA evidence, appear to have been widespread across Asia and into Southeast Asia, and produced a model showing that the interbreeding event likely occurred in Southeast Asia or the islands between the Asian mainland and Australia, as the first modern humans moved along the coastal route toward Oceania. Those migrants picked up Denisovan DNA during their transit through Southeast Asia. Then, they continued to Australia and New Guinea, carrying that Denisovan ancestry with them into isolation. Populations that moved north and west, the ancestors of East Asians and Europeans, left the zone of Denisovan habitation before or after the major admixture event and carried much less Denisovan ancestry as a result. After processing every regional population, the AI ran its final global comparison. This was the analysis that took the longest. The comparison of every population against every other population globally, simultaneously, looking for unexpected genetic connections that regional analyses might have missed. It found three. The first unexpected connection. Native American populations and Australo-Melanesian populations share a genetic signal, faint but statistically robust, that the AI traced to a common ancestral source approximately 15,000 to 20,000 years ago. This signal, previously identified in small Amazonian tribes, is detectable at low levels across a wider range of South American populations than previously documented. The implication: a population related to the ancestors of Aboriginal Australians and Papuans made it to the Americas, either via a Pacific route or as part of the original Beringian founding population, and left a detectable genetic trace in the deepest layers of South American ancestry. This was not the main founding population. It was an earlier wave, almost completely absorbed. The second unexpected connection. Populations in Iceland, one of the most isolated European populations in the world, show a mitochondrial lineage called C1 that is otherwise found almost exclusively in Native American and East Asian populations. The AI dated the entry of this lineage into Icelandic populations to approximately 1,000 years ago, consistent with the historical Norse contact with North America during the Viking Age. The genetics confirmed the sagas. Norse explorers who reached North America, Vinland, brought at least one indigenous woman back to Iceland. Her mitochondrial lineage has been passed down in unbroken maternal succession for 1,000 years and is present in approximately 80 to 100 modern Icelanders today. The third unexpected connection was the one that generated the most debate.